Bio 121, we are now looking at the three structural isomers of hexose monosaccharides. And that must sound scintillating when you actually turn on for the first moment. And what I wanted to do right at the end of the last slide was say, oh look, we can fulfill all of those rules, putting everything together from the first two chapters, thinking about what it was when you were walking around in the plains, reaching your hands up to eat a piece of fruit, fructose. Okay, you ingest it, but somehow what your cells actually need is glucose. And we'll figure out how that actually all happens at some point in time. But notice here, you count the carbons, and you should get into the habit of learning how to do that. 1, 2, 3, 4, 5, 6. What? 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. And notice, if I was to say, let's play the game we used to play in like grade school, how are they different? Notice the carbonyl structure is here on the second carbon as opposed to the first carbon, and we could go on and on and on. If this is a chemistry class, that's what you'd actually have to learn how to do. But it's a biology class, so you need to know fructose is part of sucrose, fruit. Okay, galactose is part of lactose, milk sugar. And glucose, of course, is the intermediate energy source for our cells. But notice they all have the same structural formula. So you should be thinking to yourself, how does Mother Nature tell them apart? Well, remember, one is an isomer. Galactose doesn't look like glucose to the cell. Just because you can't see the difference doesn't mean the cell can't see the difference. Fructose, notice where that carbonyl is. Notice I made the point. If this is the first carbon, that's attached to the second carbon. Okay, so that means Mother Nature can see that, even if you cannot. So what's the big deal then? You're like, oh, who cares? And you'll probably say a lot of that this summer, except you should probably learn this stuff because it's really important. Okay, so in some situations, these are long chains can form ring-shaped structures. And in fact, that happens quite a bit. Notice glucose over here. There's an alpha form and a beta form. It's the dextrose form that your body actually uses. The alpha form isn't used. But glucose itself, by doing that isomerization, you can get these multiple types of isomers. Notice that ribose and fructose look a lot alike. In fact, ribose is the sugar we're going to use to build many of our nucleic acids inside of our body. But fructose is something we convert into glucose so we can then use it as energy. The whole point is this. Rings can form on the side and it's closed and it's locked into an alpha or beta position. And then they form 5 or 6 membered rings, and these then actually will then be used for multiple responsibilities. Some of them for energy, some of them for information storage. Okay. Now, disaccharide formation, we already went through this. We went through that this is going to be a dehydration synthesis. It's going to give us a specific type of bond, which is a covalent bond. And now you're going to notice that the further we go down this rabbit hole, you're going to see that that covalent bond actually is called a glycosidic bond itself. And it results in a 1-2. Here's our 1, here's our 2, glycosidic linkage. Now that's not all that important inside of this class. It's important to understand that people have characterized that this is what occurs, but we got some disaccharides, BFD. Now other common disaccharides can be maltose, lactose, and sucrose, right? And they all form via that covalent glycosidic linkage here, you can see here. Polysaccharides, that's when things start to become interesting again to us, right? And here's glycogen stores in spermatozoa of a flatworm. And you're thinking to yourself, what is a spermatozoa using with glycogen? Well, that energy that's stored as glycogen would be used by the spermatozoa to travel, and you'll see how that happens. Now, what we do is you join together or use multiple glycosidic linkages or covalent branches, so you end up creating polymers. And the major polymer that we need to make sure that we think about is, I don't know why they used amylose and amylopectin. Okay, this is used to build multiple complex structures. If here's amylose here, here's the amylopectin, and you can see what happens is by branching it, you increase the complexity of their structure. Plants, on the other hand, will look at this structure that looks a lot like glucose, right? And then all of a sudden you start slamming it together in slightly different ways, and all of a sudden you end up with glucose. And it does it by that are linked in unbranched chains that are 1,4 glycosidic linkages. Like, I don't really care. I'm not going to ask you if it's a beta 1,4 glycosidic linkage. What I am going to get you to understand is that all cell walls of plants are made up of cellulose, and that cellulose is made up of a polysaccharide. That's what's important. Now, how do we actually use this? Well, if you think about an exoskeleton on an insect, and you may or may not understand what that means, if you have an endoskeleton, the bones inside of your body, plants, well, plants do as well, okay? But in this case, the insect, okay, the arthropod, in this case, a honeybee, okay, actually has an exoskeleton that's made up of carbohydrates. Okay, notice they're somewhat waterproof. You can see that what happens is they're flexible depending upon how they're built. And the major material here is chitin. And what they do is they throw in a little bit of nitrogen, and that nitrogen creates some flexibility here at these linkages. Now, it's important to understand that we don't just think about the biological forms of some polysaccharides, which, if I'm here, I'm thinking it's great to tell you about plants, but the reality is for mammals and for humans, when you put polymers of glucose together, let's go like this, let's go like that, let's say glucose, I'm going to use GLU for glucose, right? I'm going to put multiples of glucoses together. I'm just going to add them all together, right? And really what you end up with, of course, is a polymer of this, and that polymer, of course, is going to be glycogen. Okay, glycogen. Now, why am I stressing this? Well, you may or may not care about a plant. Maybe you're thinking I'm going to become a vegan and I'm going to eat all of this, and that's what helps the food move through your body. But even then, whatever energy has been extracted from whatever it is you're eating and converted to glucose, and that glucose reaches a specific concentration inside of your body, right? Once you reach that concentration, the excess is stored as glycogen inside of your body, okay? And that's a really important idea. Mother Nature needs to make sure that she protects the reservoirs of energy when they're available to her. Now, when we're thinking about the next type of macromolecule, lipids are those. Lipids are a diverse group of non-polar hydrocarbons, and because of that, they are hydrophobic, and because they're hydrophobic, it means we call them water-fearing. They just repel it. They're not afraid of it. Okay? You might be afraid of swimming. Non-polar hydrocarbons are not afraid of anything. Okay? They just behave a specific way when around water. Their function, they are long-term energy stores. They provide insulation for both plants and animals from the environment. They are building blocks for many of the major hormones that are used in signaling across plants and animals. And likewise, they make up these really interesting, diverse structures when you think about what it means to have a piece of fat on your chicken versus the oils that are produced onto your skin. Or how about the wax that's made to protect your ears from insects crawling into your brain? And the fact that all of your cell membranes will be made of phospholipids that you're ingesting every time you eat anything that's made up of a cell. Likewise, a moment ago I said that there are some really important hormones that are built from lipids. Those steroid structures are going to have a canonical set of physical representations that you need to understand that Mother Nature then uses to turn into signaling molecules inside of the body, regardless of the body type. So, fats and oils, okay, glycerol and fatty acids, this is how we build the major structures using ester linkages such that we can make a molecule with water. And if I told you this and we went back to how it is we built carbohydrates, you would tell me that this is a dehydration synthesis reaction, right? Glycerol molecules are attached via fatty acids and an ester linkage. So how does this actually happen? What you can see here is here's glycerol. Here's a fatty acid. Notice we're not telling you which fatty acid it is, but notice what you see here. An OH group, a carbonyl group, multiple carbons, right? But then when you add them together sequentially, here is your now glycerol backbone. Here is the different fatty acids. And what they left out of here, of course, that I will put in for you is going to be plus 3 H2O molecules. Right? That's really important. And that's important because the only way you actually make this is if you actually react. You remove these two pieces here. Okay? Cool. Now, saturated and unsaturated fats are really important in terms of our understanding of how these nonpolar tails move around one another. And you're supposed to have, I should say, Mother Nature has designed it so that you actually look at, or you have a ratio of saturated and unsaturated fats. Stearic acid, which is shown here, is a saturated fatty acid, which means, look, notice that there are no double bonds here. There's a carbonyl bond there, there's an OH bond there, but there are no double bonds here. And that means they pack tightly. And at room temperature, they may actually exist as a solid. You see that in butter, fat in meat, so on and so forth. Okay, if you think about the salami inside the refrigerator, you know, you're thinking to yourself, when I look at that fat, that is a saturated fat. Now, it may be associated with cardiovascular disease and should be limited in your diet. I'm not here to give you dietary advice. Okay? I'm here to make sure you understand when you look at a piece of fat and you see that it is solid at room temperature, you understand that that means it's saturated. Okay. Now, unsaturated fats. Well, guess what? Oleic acid, of course, is an unsaturated fat. They have at least one double bond between carbons. And what that's going to do, of course, is going to change the geometry of this structure. And that means that on the whole, they're liquids at room temperature and they're referred to as oils. If you think about corn oil or safflower oil or any of these, okay, monosaturated would mean that that cooking oil, okay, has one double bond inside of all of the fatty acids tails that are found inside of it. Polyunsaturated, which seems to be, at least the more you ingest of it, seems to be more healthy for you. Okay? Like I said, I don't really care that you remember what's healthy and what's not healthy. I expect that you understand at room temperature that this would be an unsaturated fatty acid. Okay. There are essential fatty acids. You need them. That's all there is to it. The building of your central nervous system is centered around the availability of omega-3 fatty acids. It's an essential fatty acid. It's required, but it's not synthesized by the body, which means you must ingest it from your diet. And the two major ones are omega-3 and omega-6 fatty acids. Now, this is important. This is important because you don't ingest these, you don't have a sound functioning nervous system. The interesting thing here, of course, is that they're heart healthy. I think this is a public information thing. Reduces heart attacks, triglycerides in blood, and lower blood pressure. Okay, good, whatever. I'm more interested in making sure you have a functioning nervous system, and that means you need to ingest these. Now, waxes are cool, all right? If we were in class, I'd be like, now go take your finger, put it inside your ear, tell me what you pull out. And of course, now you're going to the doctor, they tell you not to stick your finger inside your ear, but waxes are long chains of esterified alcohols. Okay, and what happens, of course, is they're hydrophobic, and they form a sticky surface. And what happens is, even though I'm thinking of you thinking about what's doing inside your ears, and notice no other place inside your body, okay, they're found on the feathers of some aquatic birds, the surface of leaves of certain plants. These waxes act for not only repelling water, but also trapping water inside spaces. Now, a major type of lipid that we need to understand is going to be a phospholipid. Okay, here's our carbon backbone, 1, 2, 3, right? So there's our glycerol backbone. We're going to attach two fatty acids to it instead of the 3 for our triglyceride. And then what we're going to do instead is we're going to put a phosphate group. And notice this is charged. And this functional group now will change the behavior of this entire molecule. And what happens, of course, is now this gets a big fancy name. If this is hydrophobic and this is hydrophilic, it means collectively we call this amphipathic. Amphipathic. All right. So then we have the hydrophobic nonpolar region here, which could be either saturated or unsaturated fatty acids. The hydrophilic top here, made hydrophilic primarily by the phosphate group there. Okay? And now what's going to happen, of course, is we're now going to use these to build something, because otherwise you're just looking at 2 circles and 2 legs, right? These phospholipids, here's some space-filling models for you can see how they all interact with one another. What we do, of course, is we put them tail to tail, heads on the outside, and now water can interact here. H2O here, and H2O here. Right? And that phospholipid bilayer now becomes a membrane. And even though we teach this to you as though it becomes part of the plasma membrane, phospholipids are used to make every membrane inside of a cell. So even though we haven't necessarily gotten to how it is we build cells just yet, okay, you're going to take these and imagine, if you will, we have this thing that we call a nucleus. Well, it has its own nuclear envelope, and that nuclear envelope is its own mixture or ratio of phospholipid bilayers. And over here will be our rough endoplasmic reticulum, and maybe our Golgi apparatus, and heck, even a vesicle, maybe our mitochondria, right? The whole point is that each one of these is a unique construction of phospholipids, and that's how the cell knows which, in this case, compartment is being made. Another type of lipid is this thing that's called a steroid. And these are important for us to be thinking about in the sense that as we're finishing up, or at least getting towards the end of what lipids are doing, notice what we've done is we've cyclicized this structure just like we did with our carbohydrates, which means we can, through chemical reactions, take any series of fats, acids, create functional groups, attach them in random places, right? And then you have the basic building block for all steroids, and that is cholesterol. Cholesterol is the major material that's required for you to actually build steroid-based hormones inside of your body. These are water insoluble. They require a transport protein to travel inside of your body, likewise inside of plants. But here's the idea. Notice what's going to happen is, say your body needs a signaling molecule called cortisol. Okay? Cholesterol is going to be modified to give us cortisol. So notice what's happened here. We switch the double bond location. Notice we put an OH over here. That methyl group stays exactly where it is. Now we get rid of this over here and we put in a carbonyl group, this other methyl alcohol and OH group there. And lo and behold, now you have a signaling molecule that allows your cells to go, hey, something's going on inside the body and we must respond to it. Okay? Cool. Now, they are hydrophobic. They're insoluble in water. It says synthesized inside your liver, right? Primary place of synthesis is for cholesterol is inside your liver. It's a precursor to other molecules, not just cortisol, but testosterone, estradiol. It's also a precursor to vitamin D, and it needs to be modified by sunlight, but it's also a precursor to bile salts, and these bile salts are emulsifiers of fats inside of your body. Okay, break them down into smaller and smaller structures. That's a big 10-penny word for you. We'll come back to it when and if we ever get to the digestive system. Now, this is a good place to pause because I want to just stop in like 3 seconds thinking about if what we've done is covered carbohydrates, now we've covered steroids, we now have 2 more groups to go, and I'll see you in a few.